So without variation, evolution is impossible. And variation comes from a couple of sources. One, mutation that we've already mentioned. Point mutations and insertions, deletions, those things we learned about in Bio 1. Those are those simple mutations. They can result in neutral variation, which means maybe there is a change to the DNA, but it doesn't give a benefit or it doesn't hurt the organism. Mutations can be more complicated in the case of like duplications, where you get extra copies of genes. Duplicated genes can then even take on new functions. Your book talks about how duplicated genes lead to more sense receptors in our noses and things, for example, during our evolution. So mutations can give rise to new alleles. That's what I really want to hit home here. So new proteins are being made, new phenotypes are being made, new alleles are being made. Sexual reproduction includes things like crossing over, independent assortment, random fertilization, those things we talked about in the chapter on meiosis and reproduction. These don't create new alleles, but they make new combinations of alleles. So some of your mom's alleles are now with some of your dad's alleles in your offspring, for example. Okay, so that's where alleles come from. New ones from mutation, from errors in copying DNA during the S phase of the cell cycle, especially when producing gametes. And sexual reproduction makes new combinations of alleles. All right, so what? Well, let's tie this back to evolution. Evolution can be measured as a change in allele frequencies in a population over time. By frequency, I mean how common is that allele. So for example, if we look at flowers again, and there's CP and there's CW, if 50% of this, so there's the color gene, if 50% of that gene in the population is CP and 50% of that gene in the population is CW, then the frequencies are 0.5 and 0.5, okay, for those allele frequencies, okay? So it's vital that you be able to tell alleles from genotypes here. Allele frequencies are those individual variants of the gene, right? So allele frequencies. Now, in a population that does not have any evolution happening, it will have the same allele frequencies every generation. They will stay the same. They won't have any changes. And we could follow a hypothetical population over time to see if it's changing, but we humans don't live that long. We need other tools to look for evolution. And this is where the Hardy-Weinberg Equilibrium shows up. Hardy-Weinberg Equilibrium is what we call that effect when a population stays the same over time. Equilibrium means the same. Hardy-Weinberg Equilibrium looks for no evolution happening, basically. So it allows us to compare expected genotype frequencies to actual frequencies in a population. Basically, Hardy-Weinberg is a null hypothesis, meaning it shows us what genotype frequencies we should expect if evolution is not happening. And by genotype frequencies, so we saw allele frequencies up here. By genotype frequencies, I mean how common is CP, 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 CW, and CW, CW in a population, okay? And we can figure out what these should be if there is no evolution happening by using the Hardy-Weinberg equation here. Now, let me break down. This is the Hardy-Weinberg equation. You should just go ahead and memorize this. It's not very hard. There's two variables in it, P and Q. If this were a Mendelian gene we were looking at, P would be the dominant allele and Q would be the recessive. We're using codominant genes, so it doesn't really matter which is which. So I'll just use, for our example, the CP and CWs that we have shown here. So P squared stands for how frequent CP will be in a population. That's CP times CP, basically. 2PQ tells you how often you should expect to see CP-CW. And then this one is CW-CW if the population is not evolving. All right. So how would you solve that? Well, basically, you just plug in you need your allele frequencies. So we have 0.5, 0.5. So you would take 0.5 squared plus 2 times 0.5 times 0.5 plus 0.5 squared equals 1. It'll always equal 1. Hardy-Weinberg equation always equals 1. Students always miss it because they think if it doesn't equal 1, it's not an equilibrium or something. But that's if you do this equation and you don't get it to equal 1, then you have a rounding issue or you did something wrong. Okay, now I set up this equation. Now what you do is you just simplify it, okay? 0.5 squared is 0.25. 0.5 times 0.5 times 2 is 0.5. And then 0.5 squared is, 0.25 equals 1. All right, so this tells us in a population where the allele frequencies are 0.5, 0.5, 25% of the organisms will be CP-CP, 50% of the organisms will be CP-CW, and 25% of them will be CW-CW. Let's look at another example. So, the idea of Hardy-Weinberg is that it's useful because we can look at a population and we can see if it is evolving at any one of its genes. Here is a population of round organisms. Each circle is an organism. This is a population of them, okay? We have 2 alleles. And I accidentally made the B's lowercase, but we have CB and we have CY. We have the Y color and the blue color allele. Now, some of the organisms are blue. They are CB-CB, okay? So I'll go ahead and write those in here. By the way, this video might come out a little longer than my 10-minute length that I like to keep them at, just because I want to get through this whole idea. Okay. We have green, which are a combination of CY, CB, CY, CB, and we have yellow, which are homozygous CY, right? Okay, so there they are. First things first, let's figure out a couple things. I want you to figure out, let me go to the next slide so you can see what we're going to figure out, okay? I want you to figure out phenotype frequencies, and then I want you to figure out the allele frequencies in this population. So phenotype frequencies are colors. So first of all, if I remember right, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 total in here to make our math easy. So blue, how many blue are there? 1, 2, 3, 4. Okay, 4. So 4 blue, that means 0.4 blue. 40% of them are blue. How many are green? 1, 2, 3. Oops, sorry, I went the wrong way. That's 30% are green. How many are yellow? 3 again, 0.3. Okay, that's the phenotype frequency of the population that we have. Okay, what are the allele frequencies? Well, here's a way to figure that out. We know we have CY and we have CB. I'm going to go ahead and write it over here again, CY and CB. Now, let's figure out how many CY alleles there are in this population. So in a yellow organism, there's 2 different CYs. So there's 1, 2 in this organism, there's 2 in this one, there's 2 in this one, and there's 2 in this one. So there's 6 there, right? Now in the green organisms, there's 1 CY. So there's 1 here, 2, 3. Okay, so we have 3 CYs here, and we had 6 here. So 3 plus 6 is 9. So we have 9 total CYs in this population. Now, CBs, how many are there? Well, we have 1, 2, 3, 4 blue organisms. So that's 8 CBs. And then 3 green organisms. So that's 1, 2, 3 more. So 8 plus 3, 8, 9, 10, 11. So that's 11 of these, okay? So what's 9 plus 11? I believe that's 20, right? Then what you do is you find the proportion. So let me put CY here and CB again. So 9 over 20 is what? 9 over 20 is 45, 0.45. And 11 over 20 is 0.55. Okay? This is our allele frequencies. Okay? Our allele frequencies are 0.45 and 0.55. Now, we need to take those and run them through the Hardy-Weinberg equation. So let me write this down. This was 0.45 and this was 0.55 for allele frequencies. We run that through the Hardy-Weinberg equation, which you can see at the top. So we're going to assign this one as P and this one as Q. So I'm just going to go ahead and solve this fairly quickly. Fairly quickly. So P squared is 0.45 squared. That is 0, so I'm simplifying this, 0.20, okay? Plus 2PQ, so that's 0.45 times 0.55 times 2. That's 0.495, which I'm going to round up to 0.5. We'll see if that's a mistake when we add it all up in a second. And then lastly, Q squared, so 0.55 squared is 0.30. And let's see if that equals 1. 0.2 plus 0.5 plus 0.3 is 1. So, or 100% of the population, right? Okay, so there is the Hardy-Weinberg equation simplified. Now, what was the point of all of this? We want to know if this population is in equilibrium, meaning is it evolving or is it not evolving for this color gene? Well, how do we tell that? Well, what we do, the way you tell it, is you compare. Let me write this down. You compare your Hardy-Weinberg results to the actual phenotype frequencies. So what we have here are predicted frequencies if the population is not evolving. So we predict that 20% of the population will be yellow-yellow. In reality, 30% was yellow-yellow. We predicted 50% should be green. In reality, 30% were green. And we predicted that 30% should be blue, and in reality, 40% were blue. So this population is not in Hardy-Weinberg equilibrium. Hardy-Weinberg EQ, okay? You compare these two numbers, and if they are not the same, then you are not in equilibrium. They have to match up. So that's how we use the Hardy-Weinberg equation. And I will have a worksheet for you to do on Blackboard to work through some of these questions.